So this is first going to be, so I called this finding probable roots for molecular evolution of translation. And finding the root is about the thinking trick, which is the first part of this. So we here are all scientists, and our business is knowing what's true. But it's amazing to think that it's only been 400 years that anyone thought this way. Only since Sir Francis Bacon, who, as you see, was a 16th century figure, you can read his quote here and what it says, and amazingly, he was saying it for the first time, that the way to know what the universe is like is to make observations and to deduce or induce from those observations. And that's a new thought, right? Humans were not evolved to think scientifically because they've only been doing it for 400 years and they're not very good at it yet, probably. But anyway, he invented it. And the conversation that you as a scientist have with the universe by making observations, seeing its true answers, it never lies. It never lies. There is no such thing as a failed experiment. It gives you the complete and honest answer to your question. And we have known since Francis Bacon that you must then interpret that answer, and that's where the trick is. Okay, but the trouble is the most interesting questions can't be approached this way. You know, if you want to think about the origin of the universe, there's absolutely no way to do an experiment. You want to think about evolution. You're a biologist, you have to think about evolution. Don't tell me about creation. You have to think about evolution. And you have no way through Baconism to bring evolution into the laboratory. Well, actually, that's not quite true. That we can now do. But you have to solve the problem of approaching interesting questions without being able to experiment on them. We can't recreate the origin of the translation system. That's what we're interested in. So how do you experiment on a topic like that? Okay, so this looks a little formidable. And usually when you try and do a development, a formal development, people just kind of glaze over and fall out of their chairs. But I want you to look at this because I guarantee you this is the trick. This is the trick of thinking that will help you think about problems like this. So take something, let's define something we'll call a sample space. This can be anything, and that's its power. We can make points in here which are where Glauco Tacchini Valentini is on the surface of the Earth. Let's say it's B. Those are his points. And over here is Silvio Berlusconi.
Ah, I don't want to be together.
Well, you're only together at this intersection a small fraction of the time. Let's say there's a reception at the Vatican, and you're both there. So now it's getting interesting.
Yeah.
Or this could be we could define what it is to say something, a fact about RNA chemistry. And this could be the RNA chemistry discussed at the course. And this could be what Dino says, and this could be what Yarus says. And there's only a tiny overlap between them, but anyway, we can define it and call the intersection. Now, here's the trick. There's a profound trick first thought of by the Reverend Bayes, and it leads to Bayes' theorem, and that's where I want to get to. There is a profound symmetry in that any two objects, and that's the power of the idea. You can build the whole of the calculus of probabilities on this kind of model. The symmetry is that there's an intersection that has, in some ways, the same relation to two bodies of facts, two bodies of observations, or two bodies of any kind. this intersection, which is the intersection between A and B, and we'll call AB, this intersection exists for both, is the same. It is the same space. It is the same set of events. It has the same probability for both A and B. One more definition and we're there. I promise you, that'll end the development. So the thing that's necessary to do is realize that this sameness, the intersection AB, allows you to do the following thing with one more idea. So from probability, so here's this set of events is A, its fraction of the space is the probability of A, normalized to everything that could happen. A happens only in there, so the ratio of these two areas is the probability of A. But we have to advance to thinking about what's called conditional probabilities. The probability of B, given that A has happened, right? So here A has happened, and in this little red space, A has happened and B has also happened. Silvio Berlusconi is shaking Glauco's hand and asking to teach at the RNA course, right? That's an intersection. He's probably too busy right now to teach at the RNA course. Anyway, that intersection is the same. The conditional probability that B has happened, given that A has also happened, is the red area. It's the intersection. And so the probability that B happens, given that, that's what the vertical bar means, probability that B happens, given that A has happened, or that A happens, given that B has happened, is in both cases related to this intersection. And you can use that to equate these other probabilities, the probability that A happens, B happens, or B, how probably that you can have B given that you've just had A, given that A has happened. You can get this thing, which just results from getting rid of the intersection, which is the same in here. That's Bayes' theorem. So you're probably somewhat less than thrilled at this point. People are shaking their heads. Yes, we're less than thrilled. But let me show you. This gets interesting. Or I claim that this now gets interesting. So a nod to the Reverend Bayes, who, as this is his only work in mathematics, he did this very simple, one step from the definition of conditional probabilities, and then he died. And his friend presented it before the Royal Society, and as a result, he has undying fame. Now, here's the way to make this interesting. If A is some kind of data, and B is a bunch of hypotheses or a hypothesis, then this thing, Bayes' theorem, which we just derived, becomes something a lot more interesting to a scientist. It says that the probability of some hypothesis, given that you have gotten data that bears on the hypothesis, divided by the probability that the hypothesis had before you got your data. In other words, we're talking about how you update what you believe on the basis of data, and that's the power of the idea. That is equal to, it is obligatorily equal to the probability of getting the data, given that the hypothesis is true, divided by the original probability of observing the data without reference to any of your prior beliefs. Okay, so this is still a little complicated, but it's beginning to be interesting. How you determine what the interpretation of what you see is all bound up, a lot of times, with Bayes' theorem. So I want to talk just a little more about that by putting it into words. So what we're talking about is, how seriously do you take what you observe, and how much should it affect? There is a mathematical rule for how much it should affect what you believe, and that's what we're talking about. one way to put it into words is the hypothesis that predicts the observations you can actually make is the correct one. And that sounds trivial, but it is not trivial. It sounds like common sense, right? But it goes way beyond what you can do with common sense only, in that it is obligatory. We're talking about a mathematical relationship that unless you want to disbelieve the calculus of probabilities, you have to believe it. It is quantitative. It is quantitative. You're going to assign real probabilities to your beliefs using this. It allows you to combine independent experiments, because everybody knows how to combine the probabilities for independent events. But most important, it means that you can study parts of what you're interested in, parts you can bring into the laboratory. You're never going to know why the dinosaurs died experimentally, because you're not going to bring a world full of dinosaurs into the laboratory and kill them. That's a ludicrous idea. But you can study that event with a part that has this logical structure, and you can make strong inductions about what happened in an event that you cannot possibly observe in the laboratory. And what you do, we're talking about probabilities, what you do when you work this way on the evolution of the code, which is what we're about to talk about, is you make some hypotheses more probable than others. And my point to you is that there is nothing disreputable, there's nothing louche about working in this way. People say, oh, don't tell me about evolution. You can't work on evolution. You can work on evolution. You can do precisely the same thing in this area that you do when you study, I don't know, the diet of groundhogs. You make observations, and as a result of those observations, Bayes tells you, certain hypotheses rise in probability. That's exactly what you do when you feed a groundhog mouse chow and dog chow, and you measure how much it eats. Some hypotheses, as a result of the data, become more probable. Now, to make this, so this is a little abstract. So let's make it a little more tangible. So here is the largest glacier in Europe. It has some Swiss name, which I never can recall. But anyway, look how beautiful it is. Let's think about this glacier. So look how orderly it is. Glaciers flow at meters per day. And you can see the flow lines here. You can see how structured and beautiful this big glacier is. But what I want to talk about is these dark lines. You can see this dark line is coming off that interface with the rock. Those are rocks. And this very orderly display shows you that glaciers transport rocks. So one way, so if you wanted to ask yourself a historical, an evolutionary question about this beautiful glacier, this beautiful scene in Switzerland, you might think, oh, well, 100 years from now, we humans will have done our reprehensible thing and there will be no glacier. How would you think about, if you saw that scene, how would you know that a glacier had shaped this valley? Well, there are a lot of ways. Turns out a lot of ways, and you can combine them using the properties of probabilities. But the thing I want to talk about is the rocks, and there are rocks under the glacier moving meters a day also. Somewhere out here, the glacier ends and it drops all its rocks. And that structure of boulders standing out in fields is extremely characteristic. That, in fact, is why I asked you to think about the picture of my laboratory. I come from a city named Boulder, and the significance of the name of the city is that it's built on a big moraine, and if you dig down in your garden, you'll find you'll find 20-ton representatives of the fact that the land that the city is built on is a big residue from a glacier that transported rocks, and then when the weather was too warm, it dropped them all in one place. So if you want to know a glacier has been working locally, you could say, well, if I find a moraine, how much does that increase the probability that I'm looking at glacial terrain? What Bayes tells you is that's equivalent to asking, how much have I increased the probability of seeing a moraine when I know that a glacier has been at work? In fact, this number, each side of this equation is much greater than 1 because moraines are not everywhere. And you, in fact, if you wanted to, you could take satellite photographs and you could measure the amount of the Earth's surface that is a moraine, right? And that would tell you that a moraine is a very good way of knowing that a glacier's been around. And that's the point. You could not only increase the probability that you're looking at a glacier valley by looking way out here at something that at first seems unrelated to the question of what's the geological history of this valley, but you could increase the probability a lot. And furthermore, you can even measure the probability. This tells you that, if you knew this ratio of areas, you would know. You would be able to quantitatively say how much your belief has increased in the hypothesis that this is a glacial valley or that there was a glacier there at one time. Okay? So you may be saying, wow, what kind of course is this in Trieste? I've come 7,000 miles to hear this kind of, never mind what it is. But I've come 7,000 miles to hear, that's common sense. Yes, it is common sense. But you can now go beyond it in a quantitative sense. You can now combine, you now know how to combine, for example, the observation that the rock floor of the valley has been scoured with the observation of a moraine. You know a lot that you did not know before, and can proceed in a much more logical and quantitative way. And that's my point. It is beautiful, though, isn't it? So in the same sense that a moraine is a non-trivial confirmation that there was an ancient glacier, we can now go looking for molecular evidence for things that we have no way to study directly, and that we might have to look far away to get evidence for. So now I want to switch to the biology. So in studying the code, we had a hint from a natural, what some people call a molecular fossil, what I now think of as a molecular fossil. It's the work of Eric Christian, now a long time ago. In that, we found that that arginine was bound by the self-splicing ribozyme, the self-splicing RNA enzyme that's a natural, it's an evolved way of maturing ribosomal RNA in the protozoan tetrahymena. This is the discovery of Tom Cech and his lab. And arginine, it turned out, was bound in the site for guanosine, and that site for guanosine was on 3 nucleotides. The blocks are supposed to be what Dino was calling the Hoogsteen side of 3 successive nucleotides, and those 3 successive nucleotides were the sequence AGA. So AGA is an arginine codon, and the fact that arginine can be bound to a conserved sequence, that's its own codon, suggested that maybe that was something general. Maybe that's something general. Maybe the coding triplets can be associated with their cognate amino acids in some way that reflects that natural hint from the tetrahymena self-splicing ribozyme. Maybe the maybe the code table is related to the triplets in its site, and and we can study that by studying the frequency of coding triplets in the sites for amino acids and seeing if that's related to the code table. In fact, not only can we study that, what this tells us is it's the same question. The two not only have the same content, they're quantitatively completely the same. And so this otherwise inaccessible event you can study in a Bayesian way by asking about amino acid binding sites and whether they contain coding triplets. Well, there aren't, unfortunately, the tetrahymena is the only natural example. Well, oh, no, it's not actually. No, now there are other examples. But you don't have very many examples of RNAs that bind amino acids, so you have to make them. So just briefly, you can go out and select new RNAs that bind amino acids by starting with a complex mixture of, say, 10 to the 15th randomized sequences that you make off random sequence DNA by transcription. And you'll look for the tiny little fraction with the red star here that has some property that enables you to grab it and purify it from the other sequences. So one cycle of that, and you re-replicate the ones that apparently work biochemically, one cycle of that won't purify it because 1 in 10 to the 15th means you need a 10 to the 15th fold purification. So that's a lot. So you have to cycle and recycle. But after a while, you can clone something that's a 100% pure new activity that RNA can do, but you didn't know that it could do it until you started it out and looked for it in this sensitive way that's called selection amplification, or sometimes selects. And then we want to ask a very simple statistical question that any person who's taken a statistics course knows how to do in a very powerful fashion. Outside the RNA site and inside the RNA site are so you've selected this to bind some amino acid. What is the frequency of the triplets? So here's something that binds isoleucine very nicely. Incidentally, some people didn't think RNA could bind isoleucine. Isoleucine has a side chain that's essentially like gasoline. And so some people said, there's some amino acids RNA will never bind. And this was on that list. But anyway, this RNA binds isoleucine. These nucleotides, in a sense, I'm about to explain to you. The lowercase ones are outside the site. The uppercase ones are inside the site. And there are isoleucine triplets outside and inside. And if the hypothesis that the code came from sites, like the arginine example, is right, there ought to be more inside. And, you know, there are absolutely straightforward ways to answer that question. You do have to define binding site nucleotides. So anything that's conserved in what started out as randomized molecules is probably a binding site nucleotide. And then there are other ways to define binding site nucleotides. For example, if you, so here this thing with an L on it is the ligand. Some small chemical reactant will react with nucleotides unless the ligand is bound, and then that nucleotide is not essential. So you know that the starred nucleotide is in or very near the binding site. Not only can you protect things when the ligand binds, you can make them more reactive. That also has to be close to, or it actually turns out, and you don't have to argue that it would be close. There are in many cases of known structure where you know that only the nucleotides that are close to the site have this property. There are other tricks like modification interference. So that's something that if you modify it before you present the ligand, that modification prevents the site from working. So anything that has that property is probably in the site. You can smash your isolated molecule with mutations And then from that mutagenesis, you can reselect things that work. The site can't change. Everything can change. So by that sort of negative criterion, where you don't have change is the site. That's doped reselection, where this is the doping with mutations. And this is the defining event, the reselection. So now we've got a big library of sites. Here's a part of it. These are some isoleucine sites. But the important thing to know is we now have, you got to do some work to take independent RNAs and define the sites in this way. So we haven't done them all. But what we've got is 337 independently derived sites for 8 different amino acids, about 18.5 kilobases, of which 5 kilobases are in sites by these chemical definitions we've just talked about, and some others that are not completely analyzed yet. So now we can ask the question, the Bayesian question. Okay, so you're not supposed to read this, but I put it up because, what the last slide seems a little insubstantial. So I put this up because it's a complicated looking thing that reassures people that we actually do have the data and we've done the analysis. So every color is a different amino acid. And so we have here arginine, glutamine, histidine, isoleucine, leucine, phenylalanine, tryptophan, and tyrosine, and we've got big sets of sequences for each of these. And out here we've got the codons and anticodons that might be in the site, and here we've got the probability of the outcome. And these dark things are things that are very, very improbable outcomes, where coding triplets end up in the site. For example, the isoleucine case that we started out talking about, the probability that the codons would be there in the frequency that we find them is 4.8 times 10 to the minus 109th. So they're improbable outcomes here. There are 3 codons and 7 cognate anticodons that recur disproportionately for these 8 amino acids that have been tested. And I've skipped over a lot of things. I hope there are questions that will enable me to fill in somewhat. But if you want the whole story, there's a Journal of Molecular Evolution review of all this data that's fairly recent in 2009 that you can read. So not only can RNA specifically bind amino acids, that was in doubt at the start. I mean, many people thought, well, we could get the code. We can see how the genetic code would arise if RNAs could specifically bind amino acids. But no one had ever observed that. And certain very insensitive experiments had not detected it. But now we know that you can bind them. And sometimes, counting all cases that are favorable and all cases that are unfavorable, we now have probabilities of random occurrence of triplets in sites that go down, taking everything into account, to orders of like 45 orders of magnitude improbable for both codons and anticodons. So not only do they bind, but they have this property. But there's yet more. There's a more specific argument you can make that RNA is doing something impressive here, that RNA chemistry, simple though it might be, based on only 4 kinds of residues, A, U, G, and C, as it is, it's still doing something extremely sophisticated. And that's an argument from simplest sites. So let me go rapidly through this, because I think my time is almost up. How much time do I have?
No, you finished, but I think you can go another 10 minutes.
Another 10 minutes, okay. So I can do this in 10 minutes. So suppose you think about some kind of primitive situation where you're making RNA. You're making it on a hot rock 4 billion years ago. you're thinking about the foundation of the RNA world, it's very expensive to make big things because to find 1.66 specified nucleotides, you have to look at 10 times as much RNA. So if you want to find something that's 16.6 nucleotides long, you got to look at 10 to the 10th times as much RNA to find that sequence occurring at random. So it's very tough to find big things. So it ends up to be very important to know what are the smallest things that are easy to find, because the things that are smallest are also absolutely, without question, going to be the most frequent things when you're making RNA on a hot rock instead of by templated synthesis. And we have a way to look at the simplest things. We make randomized RNAs that are shorter and shorter in their randomized regions, and the selection that I told you about when I was talking about selection amplification, that works and it works and it works, and you get to something that's so short that it stops working. And then you know that the thing that was working in the shortest sequence that works is indeed the smallest thing you can make out of ribonucleotides that can do the function you were selecting. Now, that also ought to be the most frequent kind of site when you've got lots of room. And so that double confirmation is a pretty convincing, I think, pretty convincing argument that you've got the smallest thing. And so we've got the smallest sites for now 4 amino acids. This says 3, but as you'll see on the next slide, it's 4. And here are the smallest sites for, by that criterion, for isoleucine, tryptophan, and histidine. And in those smallest sites, you always found, now 4 cases, arginine has also been done, when that kind of shrinking selection has been done, in 4 cases out of 4, the 4th being arginine. So here's the smallest site for arginine. Out here you have constant sequences, and the site is just this structure out here. So these are sequences we put on so we can amplify the RNA. So the site is out here. And in 4 out of 4 cases, when you do this narrower kind of selection that very specifically looks for what is the structure that evolution was most likely to encounter if it were to select something that binds this. 4 out of 4 cases, these sites, these most probable evolutionarily significant sites, have coding triplets. So not only can RNA specifically bind amino acids with biochemical affinities, they could bind them from dilute solutions, but those structures have an unusual frequency of cognate coding triplets, and these tendencies are not avoided, they are in fact enhanced when you ask the question in the most likely evolutionarily significant sites. So then the question arises, how could we use this information to envision a primordial RNA translation machine? Is there a possibility that you could build a translation machine from just what you know now about RNA and its ability to fold using the fundamentals that Dino talked about into sites for individually specific amino acids, the different shapes here? And so just to illustrate that you can carry this thought further and study it, I'm going to talk about the DIRT hypothesis, the direct RNA template hypothesis. A DIRT, or a direct RNA template, is a series of sites for activated amino acids. This would now form a peptide, the idea is, with the sequence round, square, triangular. And it would do that reproducibly. So with only activated amino acids and a piece of RNA, you could start making coded peptides. And there's a way to envision that, giving the modern translation apparatus. And something like a DIRT can exist. Here's a structure that binds, a selected structure again, that binds the peptide his phi, and it doesn't bind his or phi very well, but it binds the peptide pretty well. And the reason for selecting this is that some people say, when I talk about DIRT templates, they say, horse pucky. You're never going to have a structure that can bind two amino acids at atomic bond distances from each other, because that's too close. You can't put a his site and a phi site together, because those are big structures, contain 15, 20 nucleotides. They won't allow the two amino acids to approach so that they can form a chemical bond, only an angstrom and a half long between them. And so this is an experiment which was done by Rebecca Turk to show that that's not true. In fact, the part of it that relies on a sound molecular intuition is true. RNA does not do this. When you do this select, you have to do a complicated selection to get out the real peptide binding RNAs, which I'll tell you about if you're curious about how you avoid certain artifacts. But anyway, so Rebecca selected this, and it does not do it by making a his site, which we know how to recognize because we've done that selection. It does not do it by making a phi site. We know how to recognize those, and putting them together. Instead, it gives you an entirely new class of sites that specifically binds the his side chain, specifically binds the phi side chain, and has them at molecular distances that allow a covalent bond, because of course the covalent bond was in there in the his phi ligand that we selected affinity for. So you can make something like a DIRT. This experiment was done to show you can make something that has the properties of a direct RNA template out of RNA. You can select it. You can select lots of them. We have, I don't know, we have 8 or 9 classes of these now. And we'd like to know, of course, if they actually can not just bind something in which a peptide bond has formed, that's the product of the reaction. We'd like to know if they can catalyze the reaction. And those experiments are underway. And that's the end. And I'd be happy to answer any questions.